Hello and welcome back to the channel everyone. Hope everybody is doing absolutely fine. So in this particular video, the topic which I will discuss here was highly requested by my students following me on Instagram page. So here is the solution. So I hope after watching this video, all your doubts will be cleared for linkage and recombination. So if you like it, please share it and subscribe to the channel as well. So let's get started. So we will try to understand how linkage was observed by T.H. Morgan while conducting his experiment on Drosophila melanogaster. Now, T.H. Morgan used sex-linked genes to conduct a study along with his co-workers. So there was a team with Morgan who conducted this experiment, and the pattern that Morgan followed for this experiment was similar to the pattern of Mendelian dihybrid cross. And in this case also, Morgan selected two traits or characters for his experiment. And Drosophila are they are commonly also termed as or known as fruit flies. And the two traits which Morgan selected were for female. For female, Morgan selected yellow body and white eye, and in case of male it was brown body and red eye. Now one thing more I would like to tell here, that the brown body and red eye, they are of wild type. So wild type are basically dominant in nature. And how we represent it? We represent it as small y and w for yellow and white. All right, for female, yellow and white, because two traits we are selecting here. And in case of male, we write it plus in the subscript. So it is brown and red. I hope this much is very much clear to you all. Now what happened for F1 generation? The two genes did not segregate it independently. So what Morgan observed in F1 generation, that the two genes or the two characters did not segregate it independently, and the F1 generation are having parental type gene combination. So this point is very important. So in case of F1 generation, what had happened? No segregation and parental type in case of F1. But in case of F2, after crossing over, so after crossing over, he observed there were some recombinant formed, but the recombinant that are formed were very less in number, and he calculated that there were only 1.3% recombinant, and rest all are parental type. All right, rest all were parental type. So we will see here. All right, so rest all were of parental type. And he concluded that the parental gene combination was because the location of gene on a same chromosome were too close. Suppose these are the two genes, and we can see that these two genes on a same chromosomes are closely located, and these closely located genes are getting transferred into F1 and F2 generation more easily, or we can say that more frequently, and forming parental type recombination. So in these type of closely related genes, the recombination is very less, as genes on chromosomes are very tightly linked. So we can see here closely located, or we can say that they are tightly linked, and they form linkage. This is how the terminology of linkage was given. So here we can see that Morgan attributed this due to physical association or linkage of two genes and coined the term linkage to describe this linkage association on gene on a chromosome. So what we can see here, that on a chromosome, the two tightly linked or two tightly associated genes are getting transferred to the next generation and forming a parental combination. And we got the ratios of 1:3 recombination in this case, where the genes are closely associated. In those particular cases, recombination is very less. So we can write it over here as genes closely associated have less recombinant and more parental type. Now, in another case, what happened? He selected in another case, Morgan selected white eye and miniature wings, red eye and full wings. So he selected white eye and miniature wings and red eye and full wings. So again, red eye and full wings are wild type, and we denote it with W plus in superscript, and white eye we denote as W and M. So these are of female traits, these are male traits. And in F1, they could see recombination. Now in this case, they were able to see recombination. Why? We will understand this with the help of this diagram. So what we can see here, that the two genes on a same chromosomes, they are located at some distance. The white and miniature wings, the two genes are located at some distance, and this is the reason that the the genes are getting segregated, and we can see that there is independent segregation is occurring. This is how in Crosby we can see that recombinant type are 37.2% in Crosby, because the two genes are located far from each other and they are able to get segregated. So in this particular case, what we can see, that loosely associated genes are having more recombination, or we can say that genes which are distant apart on a same chromosome, or we can say genes that are distant apart are forming more recombinant. So in this cross B, the Morgan was able to see Mendelian dihybrid trait at some extent. So this is what happened in case when the genes are located far apart. And in F2 generation, we had seen that he observed that 37.2% were recombinant type and 62.8% are parental type. And this is what we say that loosely linked are having higher recombinations, and recombination generation of non-parental gene combination. So recombination, that is the formation of a new type. So recombination is non-parental type. So I hope this diagram is very much clear now. So one thing more I would like to add here is about genetic mapping. So one of the student of T.H. Morgan, Alfred Stuyvesant, what he did, he used the frequency of recombination. He used the frequency of recombination between gene pair on same chromosome as a measure of distance between gene and mapped position on chromosome. That is the frequency of recombination between two genes tells about the relative distance between or between genes on the same chromosome. And this genetic map is calculated in form of T.H. Morgan.